Let me first of all acknowledge my appreciation to my custodians of this land and to you, Auntie Carol, as elder here. Always a wonderful welcome to country and a great privilege for all of us, I think, to live on this wonderful landscape for a time. I want to share with you my delight and pleasure in being alive at this time. As I'm sure you all agree, specifically in relation to the era of genetic studies. Because when I was a very young fellow, DNA was identified by Watson and Crick and others, and by the time I became an honours student at UWA, DNA was accessible in some form for analysis, and that really opened up very rigorous tools for us to examine questions that previously were difficult, if not intractable. To the point where today whole genome sequencing is now occurring at a modest cost. So it's an extraordinary period to be alive, and I'm thankful that I have been alive over that period. The talk today is in three parts. So I want to talk a little bit about the restoration genetics that was undertaken at Kingsburn Botanic Garden up to 40 years ago, but certainly in full strength by the mid-90s. And I want to talk a little bit about what we learned from that and what we predicted would be going on with restoration genetics. So, what is restoration genetics? It's a form of genetics, so understanding inherited variation, and it's as applied to restoration. In a very simple way, it can be conceived as comparing various genetic attributes of restoration vegetation or animals as well with adjacent remnant vegetation. And the sort of things you might compare are levels of genetic diversity, reproductive outputs, mating systems, how pollen is moved around, and plant-pollinator interactions. So that's the broad discipline. You do it because if you understand some of these things, you may develop approaches that lead to long-term persistence. So Kings Park, let me start with this. In the mid-1990s, new legislation came into Kings Park, and that ended up being called the Botanic Gardens and Parks Authorities Act, which enabled Old Park to come under the hand of the administrative control of Kings Park. And we were charged to care for that extra piece of land in addition to Kings Park. And it was in need of repair and restoration, as indeed Kings Park is and was. Fortunately, after 3 or 4 years in the job of being director at Kings Park, I got through all the hurdle of convincing government that the place had been underfunded for more than 2 decades and needed a serious injection of funds. And part of that serious injection of funds that eventuated included money for science. So I ensured that we appointed a restoration geneticist, Ziggy Krause is his name, still there. And also we purchased under a capital works program a DNA sequencer, which was what you had to do in those days, $500,000, fairly serious funding. These days you just collect your DNA and send it off to China basically, come back with the sequence for the whole genome if you so wish. So we had the capacity in the mid-90s to start looking very carefully at the genetics of organisms of interest. Now Bold Park, it straddles the Quindalup dunes, right on the coast's edge, and the Karrakatta dunes, and the interface between the two. And one question we had was, if we were going to repair and restore degraded, weed-infested vegetation in Bulb Park, from whence should seeds come? Now, genetics in general terms has been driven by animal studies in the Northern Hemisphere, and studies of humans, for example. And the pattern for highly mobile animals is they tend to have most genetic variation within populations. So in humans, chances of me having the same genome as you is very remote, but very little difference as you move across landscapes. So little geographical difference in genomes. And that's become the standard model that population genetics textbooks are written about, and the restoration genetics textbooks are written about. Organisms that have most of the variation within populations and not much going on as you move across their range. So in Bulb Park, we identified 15 species that range from trees like Banksia down to kangaroo paws and orchids to represent the type of vegetation in the place. And we identified about 20 or so potential seed collecting sites of remnant vegetation up and down the Swan Coastal Plain, shown on the map. Only 100 kilometres or so from the extreme north to the extreme south. And based on traditional knowledge, we would have expected not much variation at all in all those species. So we could collect seed from anywhere up and down for all those species. That was a hypothesis based on the existing literature. And indeed, of the 15 species, we found that applied for 2. So this graph, I'll show you a few of these. They're just genetic distance axes on the x-axis and the y-axis. So if ellipses overlap like this, that means there's no difference. If there was complete difference, you would expect the ellipses to be completely separated in that two-dimensional space. So here's Mesomelina stygia. You can see the collection sites for this one, about 5 sites, predominantly on the Caracara dunes. And there's not a shred of difference. So with this sedge, you could collect seeds from anywhere up and down the Caracara dunes, and you would be sampling virtually the same genotype. This is like a human, in essence, but only 2 of 15 species. So what were the others doing? This is more than half, or half on average, like 8 of 15 species. Common red and green kangaroo looks identical in Bulb Park to those in Kings Park, to those in Midland, to those up to Lancelin and down to Mount Barker. When we looked at the genetics of it, this is what we got. And no overlap between Bulb Park and Kings Park. Extraordinary. In fact, Midland showed a slight bit of overlap. But if you look at the little map down below there, the general principle that you should become more divergent the further apart you are didn't apply. Geographic distance doesn't equal genetic distance in this. So Bulb Park is much closer to Kings Park than Midland is, yet Midland is more near than Kings Park. That was just in the nick of time because we collected tens of thousands of seeds of red and green kangaroo paw in Kings Park, and they were about to be splattered over Bulb Park. In fact, in Bulb Park, the Quindalup red and green kangaroo paw is a different one from the Caracara dunes. And this is extraordinary in global terms to have this sort of stuff, and people elsewhere would be saying, if these were birds, they'd be different species. And yet morphologically you can't pick them. So something really interesting was going on. And it got even more interesting, just as with the red and green kangaroo paws, we had a lot of Conostolus aculeata because we got serious funding from a mining company and the state government to repair and restore the Kings Park scarp. And Conostolus aculeata appears in the scarp. So just as Ziggy was appointed and the DNA machine came in, we had this massive collection of Conostolus aculeata from Nirabup, and that was about to be placed all over the scarp. And we applied the DNA test, and in fact there are 3 subspecies of this species within Kings Park. So the top, your top right is Conostolus aculeata has broad leaves, short flowering stems. Conostolus aculeata signorum has broad leaves but globular headed flowering stems that stick way above the leaves. And on the scarp there's a form like the river vole form, which has narrow leaves, and usually they're about level with the top of the leaves. Occasionally one a bit longer. And look at what happened when we looked at the DNA. Signorum in the dark blues barely overlapped with subspecies of Culeata within King's Park itself. And the same story, only a tiny bit of overlap with the scarp form of the Pricei, which is the narrow leaf, all in King's Park. And the green circle is the genotype, encompasses the genotype of the nearby population. So you can see we were going to homogenise Condystolis aculeata in King's Park because we didn't understand the genetic variation. So, deep breath. That was quite a salutary lesson. I'd worked for a long time on Eucalyptus caesea, and going right back to the days when all we had were alizymes. Caesea occurs on 25 graded outcrops in the central wheat plot, in in moderate to very small populations, and it behaves like nothing on this planet. Almost every population almost is different. So this graph, same sort of idea of just genetic diversity measure for DNA, sampling by Nicole Besmer and her PhD. Their dispersion of genotypes. So, can I use the cursor? Okay. So, can you see these green dots just here? Mm-hmm. And follow the arrow down. There's the green dot. And look at the bright blue dot. They're separated on the primary axis, completely different than many species of vertebrates, for example. And in fact, the green dot is more similar to Chewcooping, 100 kilometres to the northeast. So these were very early observations that have been reinforced. This is a paper that just came out last year now. And as the DNA tools have got better, just the story has been confirmed rather than radically changed. Something unusual is going on in southwestern Australia. Now, with restoration, likewise, if you just start thinking about reproductive biology, restoration ranges from things that are dead easy to germinate, can grow anywhere, and will come up at the drop of a hat, like Yorkum. They have common pollinators, ranging from honey eaters to mammals to insects. But consider trying to get something like this into a restoration patch. So this is a dragon orchid with its specific pollinator, a male finnered wasp that only goes and pollinates that flower. And it's also grown with growing with its own mycorrhizal associate. So the relative complexities of those two organisms in terms of restoration are profound. And if restoration is about restoring and repairing biodiversity, then we've got to address those sort of questions. We've got to understand our organisms well enough to know which ones are dead easy and which ones aren't, and why aren't the ones dead easy? What's the reason for the difficulty? So at the same time in the 1980s, with a team of people, we were looking at a rare species in the southwest, and when we published a little book in 1990, we just reviewed the available information at that stage, and it became very clear that there is an unequal dispersion of rare and threatened species across the landscape. So uplands, like the granite rock shown there, that are infertile and very old, had 2/3 of the state's declared rare flora. Lowlands, wetlands, those sort of things had just 11%. And there was 25% in between. So again, something really interesting was going on. So this was telling us we need to start considering the hills as special cases in conservation. And This body of theory was published now 10 years ago and is slowly being tested and further investigated. A simple way to express it is the big duality in our landscapes. So just opposing the old uplands and the younger lowlands. The lowlands can be coastal and they can be wetlands. Now some respond rapidly and readily to manipulation. So this is the lowlands, of course, wetlands and coastal dune systems. It's simple restoration. You can establish the abiotic habitat structure, and if you choose your species, you can really get restoration happening very quickly in these landscapes. But others with exactly the same treatment, just different species, don't respond in the same way. The species tend to have special biological traits. They're also incredibly rich habitats compared to the lowland landscapes, and they have lottery-style recruitment, community assembly. Restoration efforts are often complex, slow, uncertain, or impossible, based on present knowledge. So we need, if we're going to do restoration ecology, to understand the genetics, and the connection between the genetics and landscape is the thesis I want to present to you. So here's a photo I took yesterday, walking along the beach, good beach as I do, and typical yodful, the young, often disturbed lowland fertile landscapes, and on the horizon, Granite Hill. If you want to do restoration and have quick wins and quick successes, where would you go? Even though it's damaged and there's salt spray killing the Agonis and bare patches of sand, you go for that lowland, because those species are really tough and they can hack disturbance. All you've got to get is seed and give them a bit of protection where you go. Go up under the granite outcrop and try and do the same. I don't know anyone who's restored granite outcrop vegetation anywhere. It's just so complex and challenging in a biological sense that it will take us a very long period to do that. So, pretty simple, straightforward messages you would think. Here's this is Steve Van Nooyen's PhD done in the 80s. He worked on Banksia trichospis, a global distribution of 13 kilometres maximum length, confined to the Mount Azure uplands. And no one has worked out how to repair and restore northern Quonkam, even though we're mining sand and all that sort of stuff. The attempts at restoration and repair started off with wheat, I think, or oats, and it's advanced a little bit towards native plants. We still, I would suggest, don't have a clue about how to do that, how to get Quonkam back. We don't know how to manage fire in the appropriate way. What Stephen Van Nooyen found was it took 19 years before Banksia trichospis flowered from seed, let alone built up a seed store. And furthermore, with this paper of environmental non-seed production and mortality, they discovered tremendous pressure on the seed store, so there wasn't much seed at all. And a lot of it was to do with moth larvae that parasitise the floral axis, and black cockatoos, which with about 80% accuracy can pick which axes have been parasitised and go for it. So really complex, interesting, threatened bird caring for a threatened plant situation. So I made a series of recommendations in the 1990s about what you might do based on these observations. Pointing out that terrestrial upland systems removed from the coast and terrestrial upland wetlands pose the greatest challenge. The sort of things you might do is leave the soil surface intact wherever possible, manage weeds, beware of frequent fire, especially on sandy soils. You'd have to physically disperse propagules. You can't rely on the bush recovering itself. You would expect change, induced or otherwise. You would certainly expect failure as the primary outcome, given we know so little. And you'd need to learn through research and adaptive management. So this is the 1990s. And you think, okay, well maybe, what are we now, 25 years on, maybe things have changed. That's and it's a long-term business, fairly clearly. So I want to move on to the Gondwana project because this is a great example of cutting-edge restoration in the landscape. And the team of us got assembled. Gondwana, let me see if I can bring up this map. I just wanted the green ellipse shows you where Gondwana Link is, with the Stirling Range to its left and Fitzgerald to its right. Red colour is richest places for plant life, based on the work that Paul Jura and I did. And notice that the Stirling Range is only pink compared to the Gondwana, southern Gondwana Link area. That area is richer in species than the Stirling Range National Park in a comparable area. Salinity is shown in blue, so the saline creek lines there. Phytophthora is yellow dots, and lots of them in the Stirling Range, a couple in the southern Gondwana Link area. And you can see the vegetation is fragmented, but there are big chunks of bush. And Gondwana Link therefore seemed like a nice place to work, not only because of that situation, but because of the Gondwana Link project. So by 2014 or thereabouts, about 11 to 12 million had been spent on restoration in Gondwana Link properties. More than 3,000 hectares have been purchased and replanted in critical areas. And this is part of a larger program that's also secured 8,000 hectares of existing habitat. The original concept of Gondwana Link was founded on mega corridor theory. So it was founded on this notion that organisms are like humans, as I described right at the start. Basically that organisms move around the landscape, migrate if there's unacceptable habitat conditions, and that's what goes on. And the question that I had running through my mind, I've debated with Keith Bradley often, was, does the Gondwana Link restoration capture all or most biodiversity, and especially those bits in the uplands, the species-rich places? So that's the question, and so with Dave Coates we cooked up this project, and the thinking was fairly simple. So here's Eucalyptus occidentalis growing in a restoration site, and there it is in on the right-hand side in a remnant of native vegetation. So basically, if we compared the genetics of occidentalis in those two situations, if restoration was working, the genetics should be pretty much the same according to various measurements. So should the mating system, so should the pollination. This is a species. Anyway, that was the idea, and basically we discovered very soon that what's been established on the restoration sites is a selection of native plant species. And what do you think might drive that selection?
Price.
Price, absolutely. And price of seed, pretty fundamental. So the object of Gondwana Link was to cover area as rapidly as possible. In order to do that, you're going to need fast-growing species that produce lots of seed and that will germinate very easily. So that almost immediately excludes the species-rich uplands, where all those weird biological things are going on. Justin Johnson has been involved in this, and he recognised that the initial plantings weren't including the species-rich upland stuff, so he came up with this. See if by growing them together in small patches, that is a better mimic of what's going on in the bush, which it is on face value. So are Justin Johnson's proteaceous nodes working is a key question. And to me, this was the critical question: are there predicted differences in genetic patterns and restoration success between upland rockwool species and lowland dogwood species? That's a critical question if we're ever going to restore and repair biodiversity that is in the landscape undisturbed. So we applied to the Australian Research Council and we got funding, which is great. Dave Coates and I had a yarn one day and we resolved this would be a good project. And he said, oh, I've got a bit of money in Deepore. Linkage projects work on the basis that the federal government will match or go up to 4 or 5 times hard cash provided by a partner. So with 120,000 from Deepore and 15,000 from BGPA, we were able to get 400,000 out of the federal government. And that was enough to employ a postdoctoral fellow, a geneticist, and provide her with enough money to do her work and travel and all that. Gondwana provided in-kind support, liaison with landowners, that sort of thing, and UWA likewise in-kind support. Here's the team. We got out in the bush and had a very enjoyable day in 2015. Because these things have to come through the university, I was put down as the chief investigator. And then you can see the rest of the organisations, either Deepore or BGPA, except for Justin Johnson. So Justin and I were the only Albany people in the team, all the rest were Perth-based. And 2 overseas students also came in and were based in Perth to look at pollination ecology. So the sites, there were up to 4 possible restoration sites. Most of you probably know these, so I won't dwell on them, other than to say that they're all incredibly valuable, they're all privately owned, and have restoration going on. But they do differ. It's not an ideal experimental situation, so they differ in seed and seeding establishment regimes, different lengths of time they've been established, from 5 to 15 years, and different degrees of consideration of landscape age and fertility. So in some cases seed will just broadcast on all sorts of landscapes anywhere, upland, lowland, wet or dry. In other cases a little more attention to detail was provided. And the approach was to sample leaf and seed, to germinate the seed, often at Kings Park, extract DNA from the seed, genotype all the individuals with 10 to 12 microsatellite markers, DNA markers, assess genetic diversity and genetic divergence using adult leaf material, look at progeny arrays, that is, what seedlings are produced by known genetic mothers, and assess pollen dispersal by mapping mothers and that sort of thing. Midway through the 4-year project, I gave this summary. Basil's moving around a bit. How am I going? So that's going okay. So the summary was that we had a team field inspection and we agreed on experimental design. A fundamental observation was there weren't enough cockerel species in the restoration sites for us to test that big idea that I was really interested in. They commenced collection of material from field sites for genotyping. That was done by the postdoc who hadn't done much field work. I'll just leave that comment with you. And built up microsatellite library, superbly done in the lab. All the lab work was just classic world class. 6 species were chosen. Eucalyptus occidentalis is in brackets because it got cropped because the field seed collecting team were short. The trees were high and they couldn't work out how to seed. So the practical considerations, these seeds are the buggers, you know, you really have to get expert seed collectors in your team on this kind of thing. And then projects by the international students. Justin Johnson's PhD on ecological things was, the hope was to integrate that in pollination studies too. 5 species ended up being examined. They were from different genera and they all had good seed set, including the upward species, Banksia media. They were what we call foundational framework species that were put in to get vegetation into the landscape quickly. They were used over the broader landscape, so no attention to uplands, lowlands was paid. They're all animal pollinated and only one Banksia media is from hotblods. In this midway point, it was clear that we weren't going to address ecological questions unless Justin Johnson pulled the rabbit out of his hat. nor were we going to test the upland, lowland idea. So we just flagged that was really necessary. Extraordinary things were going on. So with the vertebrate pollinators, things like pygmy possums and honey possums were coming in within, was it 4 years, something like that? Yeah, something like that. So the vertebrate pollinators were, and some other birds were coming in very early in the revegetation. Really good success. We've had talks about that previously. Things like the black honey were recorded by Angela Sanders for the first time closest to Albany, and they're a desert species. They were coming down when the desert was in drought. So all sorts of great things were happening. But what was going on, and I'll just give you 2 examples. So Melaleuca acuminata, which grows along creeks, quite widespread in the wheat belt. And this is DST, so this is an estimate of population divergence, where 0 equals no divergence, and 1 would be complete divergence, no genetic similarities. And you have to interpret the places that REM means REM vegetation and REST means restoration at the end of each of those acronyms. But just note, of the 4 sites in Gondwana, nothing gets up to even 0.1 in DST measurements. So they're really like 0.023 or 0.55. These are behaving like humans. The genetic diversity that was in the restoration is the same as in the remnants. No problem. That suggests the seed collections were of local provenance, but may not have been, and this is a classic dogfoot species response. What was Banksia media doing? There are no seed collection records, but high divergence among restoration and nearby remnants occurred over 1.5 kilometres at Penia. And also high divergence among the remnants, which were all less than 25 kilometres apart. Is high divergence among populations of media typical? Was Melissa's question. I was like, yes, absolutely, it's an optimal species. And you were about that 25 years ago. Were seed sources in fact quite local? Yes, is the answer. So records of seed sources would have been highly informative. So is restoration working? What's the summary? Is similar genetic diversity being captured? So this is Melissa's summary. She's got a happy face here. She says yes. Genetic diversity available in remnant populations has been captured, and I'll put in brackets, but predominantly Yodophil species examined here. So we know we can do this. We can do broad-scale restoration, but you have to have species from lowland, very fecund species as the primary target, and we're lucky. Yeah, no data. There was seed source from local provenance. She's surmising probably, yes, but not in the Proteaceae, which Banksia is involved, and she's trying to cast the best side to the work. This is fundamental to papers and career. So generally a good outcome consistent with restoration aims, and I wouldn't contend with that. But 2 large caveats. Firstly, the optimal species are not tested, and secondly, there's been no recruitment at any of the restoration sites for any study species. I think that's still true.
What does that actually mean, no recruitment?
That means that the plantings that occurred, they've grown to reproductive maturity, they're producing seed, some of them are dropping seed on the soil, others are keeping it in the canopy, and not a single germinant has been seen, even with 15-year-old restoration.
The exception is Acacia dictyonura.
Oh, we've got 1 species. Excellent.
Prolific.
Okay.
And now dying is a drought.
Okay. So that's fertile and lowlandish in These are all the mating system and reproductive questions. And here's some graphs, and all I want you to take from the graphs are that the outcrossing system, which is TM, is the same in remnant and revegetated things, so the same, not statistically significant in Melaleuca acuminata, again a lowland species. The number of pollen fathers, the number of individuals in the population, they're all the same when you compare the 2. But when you look at Banksia media, less data, but in particular This is Monjumuk, remnant Monjumuk reed edge, and it's fundamentally different, very low values for in this case, the number of fathers, but completely different on the reed edge. So there's something weird going on in ancient media. We would predict that something weird would go on. So is it working? Is pollination resulting in comparable outcrossing rates? Yes, for the insect pollinated species from Gulf is not the insects are there. Reduced outcrossing may be some concern for proteaceous, I'll put it in brackets, popular species of Penia. So it's the same story. The lowlands fecund species are doing well, they've got pollinators, mating systems the same. Patterns of pollen dispersal are very different and complex, is all I want you to know about that. And there's pollen immigration coming in from if there's adjacent native vegetation in the bank. So where to from here? I've got just 2 more slides. Melissa's points first. The importance of keeping good seed source records would help in revegetation. That was a challenge for this study. We don't know how divergent populations are to augment populations in the future. Given the shortage of seeds, restoration sites could be seed orchards. And they work very well in that regard for cyclops, acacia cyclops and Melaleuca curcumina. The ecological and pollinator observations research could help inform animal pollination limitations, but not enough was done. This was an over-ambitious project to imagine we do ecology and pollination as well as genetics. And there's certainly reduced outcrossing rates in the banksha. So they're more inbred. And a traditional geneticist immediately says that's a problem. Someone who studies old landscapes says there are ways through that. It's not necessarily a problem. Eucalyptus species, we have good evidence for that. Recruitment has to happen. It might require active management. And my 2 points are that till Justin writes up his PhD, we don't know whether ecology is going to further inform this process. And secondly, we haven't addressed that big question that excited me at the start of it when I went chief investigator.